This is indeed a topic of interest at the moment, because some of you may have seen that yesterday, right yesterday, in the journal Arteriosclerosis, Thrombosis and Vascular Biology, there was an editorial exactly on this topic. So I think we are going into an area where there has been dispute around, and I will try to give you my personal view of this that is very much open to questions. These are my disclosures, mostly for speaking on topics related to omega-3 fatty acids. And I will start with these historical pictures portraying Jorn Dierberg and Olaf Bang in 1978 during one of the 6 expeditions that they had to Greenland, when the entire story of the potential benefits of omega-3 fatty acids related to cardiovascular disease was born. Things were easy, however, at that time. There was knowledge about the metabolism of linoleic and alpha-linolenic acid, the precursors of the omega-6 and the omega-3 long-chain polyunsaturated fatty acids, even with some possible oversimplification of the metabolic pathways. For example, at that time, the pathways of conversion of eicosapentaenoic into docosahexaenoic acid were not as known as we know today through the so-called Sprecher's shunt. But the general idea was at that time that the entire world, the entire medicine, the entire biology could be explained by the simple paradigm, the yin and yang of thromboxane and prostacyclin. So the hypothesis at that time was that if you have arachidonic acid in the SN2 positions of phospholipids, you have a precursor of thromboxane A2, mostly in platelets, and of prostacyclin or prostaglandin I2, mainly in endothelial cells. These two compounds are endowed with potent biological properties, namely platelet aggregation and vasoconstriction on the one hand, inhibition of platelet aggregation and vasodilation on the other. When you substitute arachidonic acid with eicosapentaenoic acid, and I will remind you that arachidonic acid is named eicosa tetraenoic acid because it has 4 double bonds, and you substitute with a fatty acid with the same chain length with 1 extra double bond, you give rise to prostanoids, namely thromboxane A3 and prostaglandin I3, with different biological properties. Because while prostaglandin I3 is still able to inhibit platelet function and to cause vasodilation, thromboxane A3 is virtually inactive on platelets and on smooth muscle cells. So we get, at the end, an imbalance in the production of vasoconstrictive and platelet-active mediators, leading to a net cardiovascular benefit. This was the hypothesis, but of course these hypotheses are oversimplification of the reality, and we know that things are not as simple as they were portrayed at the beginning. And for example, we know that the complexity of the system is increased at least by 1 order of magnitude by knowing that there are other pathways of metabolism of polyunsaturated fatty acids through the P450 epoxygenase pathway to the 5-lipoxygenase pathway. So we have also the production of leukotrienes of the 4 series starting from arachidonic acid, of the 5 series when we start from EPA. And then we heard yesterday through the presentation of Dr. Serran that novel mediators, novel pathways have been recently uncovered, and these are through enzymes that are able to produce a number of other bioactive substances, some of which arise also from what was usually thought to be an inert precursor, docosahexaenoic acid, DHA, that is able to be transformed into some of the resolvins, of the maresins, of the substances that you heard yesterday. So an enormous complexity of biologically active mediators arising from arachidonic acid and EPA and DHA. Therefore, substitution of EPA and DHA and indirectly alpha-linolenic acid to arachidonic acid in membrane phospholipids gives rise to a profound but also extremely complex change in the generation of bioactive lipid mediators, mostly with an anti-inflammatory and we heard the terminology pro-resolving mediators, preventing the initiation of inflammation and facilitating its resolution. But there is also another added level of complexity on this, that changes in fatty acid composition in membrane phospholipids can also cause direct changes in the pattern of gene expression, not necessarily mediated by the generation of active metabolites. And in the broadest conceptualization that I can have of the entire topic, we can distinguish two general types of effects, broadly definable as non-genomic and genomic effect. Non-genomic are what we have already described, the effects on vascular tone and platelet function that are to some extent explainable by the decreased production of thromboxane A2. But then we have also genomic effect, one example of which, probably the first one to be uncovered, was what we described in 1994 when I was working at the time at Brigham and Women's Hospital in Boston, in which we described the property of EPA and DHA when incorporated into the membranes of endothelial cells to confer an anti-inflammatory, anti-adhesive phenotype to endothelial cells. You see here the monolayer of endothelial cells in control conditions exposed to a suspension of monocytes or monocytoid cells. Usually in these conditions you can wash off the monocytes because endothelial cells in control conditions are inactive from the standpoint of supporting the adhesion of white blood cells, of leukocytes in general, and of monocytes, particularly relevant to atherosclerosis in particular. But when you expose endothelial cells to an inflammatory cytokine, for example, interleukin-1, tumor necrosis factor, interleukin-4, as exemplified here, or bacterial endotoxin, you have an enormous increase in the adhesion of monocytes to endothelial cells. This process is to a large extent mediated by the expression and exposure on the membrane of endothelial cells of an adhesion molecule that supports the adhesion of leukocytes and in particular of monocytes, and this molecule is called vascular cell adhesion molecule 1, VCAM1, and you can block to a large extent the increased adhesion of monocytes if you block the function of this protein with a monoclonal antibody against this molecule. You see that if you have, however, pre-treated endothelial cells with docosahexaenoic acid, which is the most potent of the fatty acids that we investigated at that time, you have that in the same conditions of exposure of endothelial cells to the suspension of monocytes, you have much less adhesion. And this is due to the dose, concentration, and time-dependent decrease in the expression of VCAM1 on the surface, which in turns depend on less transcription of the molecule, as exemplified here by the steady-state levels of messenger RNA as seen in this old northern analysis. Later on, in the following years, we discovered some mechanism by which this takes place. And here you have a gel shift, the so-called electrophoresis mobility shift assay, by which you assay a transcription factor, the activity of a transcription factor, in this case a nuclear factor kappa B, that is thought to mediate the increased transcription of the gene for VCAM1. And the intensity of this band here is proportional to the amount of transcription factor that becomes bound to DNA, forming so DNA protein complexes. And as you see here, the intensity of this band is sharply increased when you prime endothelial cells with an inflammatory cytokine, interleukin-1, and you can quench this activation in the presence of DHA at concentrations in the low micromolar range, which are thought to be nutritionally and pharmacologically relevant. So the theory around this is that when you have, in this case, the surface of an endothelial cells in which you have receptors for cytokines, for the advanced glycation and products of diabetes, for minimally modified low-density lipoproteins, or you have also stimuli such as formol myristate acetate that bypass receptors, any receptor in the plasma membrane, and simply can permeate the cell. Then you have an increase in the burst of production of reactive oxygen intermediates, most important hydrogen peroxide, which is the main activator of the assembly of these two components, P65 and P50 of the nuclear factor kappa B system. And in this case, the removal of an inhibitor, I kappa B, allows the dimer, the heterodimer, to transmigrate into the nucleus, where it can sit on consensus sequences in the promoter region of several inflammatory molecules, such as VCAM1, ICAM1, E-selectin, and some secondary cytokines, other secondary mediators. And this is the general pathway by which inflammatory mediators trigger the so-called pleiotropic phenotype of endothelial activation, by which one stimulus can give rise to a number of inflammatory molecules being produced or exposed on the cell surface. And if you can quench this activation, as you can do in the presence of fatty acids, you come at the end with an overall anti-inflammatory phenotype, which is what we observed in endothelial cells. I don't want to dwell on this busy table, but this is just a demonstration that some of these effects are probably direct effect, do not require the transformation into bioactive mediators, because you can block the various pathways of metabolism that have been discovered by several different techniques, pharmacological or small interfering RNA techniques. And at the end, you end up with an anti-inflammatory and also anti-atherogenic phenotype, because in the animal model of the LDL receptor knockout mice, which are prone to atherosclerosis, especially in the area of the thoracic aorta, as shown here, treatment with omega-3 fatty acids, as you see in B, can to a large extent prevent the atherosclerosis, and you see here that there is an important inhibition of these overall, especially in the area of the aortic arch in the animal. And this is interesting because the morphology of the plaques that occur in the aortic arch in the animal resembles quite much the so-called unstable plaques that we can see in humans in the coronary arteries of subjects dying from an acute coronary syndrome, giving rise to the complication of the plaques. However, you also see here that feeding these animals with the same pro-atherogenic phenotype with the corn oil, which is rich in omega-6 fatty acid, also gives rise to an anti-atherogenic phenotype, and you have, and we can see this better in this magnification of the graph, you have some inhibition of atherosclerosis also with the addition to the diet, or to the normal chow diet of these animals with corn oil, which is rich in omega-6 fatty acids. Therefore, both omega-3 and omega-6 exert anti-inflammatory effects, as shown by clinical data. I will dwell on this because this requires much clarification. Animal data, for example, the anti-atherogenic effects seen in the LDL receptor knockout model, and in vitro cellular systems. For example, the inhibiting effects of the cytokine stimulated the expression of adhesion molecules and of other inflammatory mediators such as COX-2 and others that we discovered and we described later on. It is likely, therefore, that both are beneficial when they substitute saturated fatty acids, which is the species that virtually invariably is associated with the largest pro-inflammatory phenotype. But would omega-3 and omega-6 compete with each other? And I see the problem as two separate but also intertwined problems. On the one hand, we have the competition between omega-6 fatty acids versus saturated fatty acids, which is what we described in the animal model when you give corn oil to the animal, and you can describe in these conditions a protective anti-atherogenic phenotype. And on the other hand, we have the problem of omega-6 fatty acids versus omega-3 fatty acids. Indeed, in vitro, coming back to in vitro experiments, when you take saturated fatty acids on the background of a stimulation of endothelial cells with an inflammatory cytokine, you have really no effect. So saturated fatty acids from this standpoint are relatively neutral. But as soon as you introduce fatty acids containing at least one unsaturation, you get some inhibition, which is maximum, notice this, is maximum for long-chain omega-3 fatty acids and mostly for docosahexaenoic acid. And you see that also some mild inhibitory effects also can be described with oleic acid, palmitoleic, ricinoleic acid, which are omega-9 monounsaturates. You see here that there is a time-dependent inhibition that can be described for DHA maximum, but also for arachidonic acid, for linoleic acid. When we try to understand the structure-activity relationship of these inhibitory effects of fatty acids on endothelial activation, the brief summary can be that saturated fatty acids have no effect on cytokine-induced endothelial activation, but a progressively increasing inhibitory activity can be observed for the same chain length with an increase in the number of double bonds. And comparison of fatty acids with the same chain length and number of unsaturation and only different for the double bond position or for the cis-trans configuration indicated no difference in inhibitory potency, and thus the double bond in our results is the minimum necessary and sufficient requirement for fatty acid inhibition of endothelial activation. So the theory is this. You have the plasma membrane, you have imagine an endothelial cell surface where the cytokine receptors are located. When you engage the cytokine receptor with the agonist, for example interleukin-1, you trigger the activation of a reactive oxygen species generating system producing superoxide anion as the first species, and then hydrogen peroxide. And hydrogen peroxide is itself the molecular species that is most likely to activate the NF-kappa B system in turn leading to the phenotype of endothelial activation. But if you have in the plasma membrane these unsaturated fatty acids, then you have a diversion of hydrogen peroxide with the formation of peroxides in the membranes, and less hydrogen peroxide that is available for activating the system, and this in turn causes a decreased activation of the NF-kappa B system. Now, you will hear more in the same session the results that can be reproduced in the animal model by Dr. Kang in the transgenic animal that has insertion of the gene for fatty acid desaturase, the enzyme that is able to convert omega-6 into omega-3 through this interesting and important technology. And in this case, you can have fatty acids that you have animals, mammals, that are highly enriched in omega-3 fatty acids in their profile. And if you take, as Dr. Kang will certainly show you later on, you take these transgenic animals that have a high amount of omega-3, an extremely high amount of omega-3s in their plasma membrane. If you hybridize with wild-type mouse that have, for example, a pro-atherogenic genotype, for example, the LDL receptor knockout or the ApoE knockout mouse, you can test whether these extremely important increase in omega-3 fatty acids in the membrane can also lead to a pro-atherogenic phenotype. And this is indeed what has been shown and what has been reported very recently, and I'm sure that Dr. Kang will expand on this with a reproduction of the findings that you can have with supplementation of the diets of these animals with omega-3 fatty acids, but also with a much more profound change in the anti-inflammatory, anti-atherogenic profile due to the extreme changes in the ratio that you can have. And this is parallel by a profound decrease in the expression of addition molecules such as VCAM1, ICAM1, and E-selectin. But I will surmise to you that the extreme change in the omega-6, omega-3 ratio observed in this model compared with the wild-type phenotype does not itself prove the existence of absolute pro-inflammatory effects of omega-6 fatty acids, because these results can also still be fully explained by the high amount of omega-3s that these animals have in their plasma membranes. Some other arguments for the theory that the ratio is relevant could be derived from nutrigenetic studies in the Los Angeles heart study by Dwyer and others reported in 2004 in humans, when these investigators determined the fact that carrying of some variants, copy number variants in the promoter region of this enzyme, 5-lipoxygenase, which is the main enzyme favoring the production of leukotriene B4, these individuals that have copy variants also have an important pro-atherogenic phenotype in terms of carotid intima media thickness. In these individuals, if you have the copy number variants compared with the wild type, you have increased atherosclerosis in terms of increased carotid intima media thickness. And in this case, if you divide these individuals according to tertiles of intake of linoleic acid, you see that there is really no change, no quenching of the pro-atherogenic phenotype in these animals, and actually in tertiles of arachidonic acid you can see an increase of the magnitude of these differences between the carriers of the common allele and the carriers of the two variant alleles here. But conversely, when you divide the individuals in tertiles of nutritional intake of EPA plus DHA, what you have in the genotype does not express in the phenotype because there is a quenching of this proatherogenic genotype by the diet extremely rich, in this case, in the highest tertiles of omega-3 fatty acids.
Please, 5 minutes.
Yes. The conclusions based on these in vitro findings and their relevance for the interpretation are that whenever you substitute saturated with unsaturated fatty acids, you have to expect some anti-inflammatory activity. This is maximum for omega-3, both because of the production of unique lipid mediators and for the direct effects of this highest degree of unsaturation. And omega-6 and omega-3 are likely allies when they compete with unsaturated fatty acids, with saturated fatty acids, sorry for this. But omega-3s are the most active anti-inflammatory fatty acids, and to some extent they compete biochemically and functionally with omega-6 fatty acids. The last piece of evidence that I want to give you, however, is that this has to be framed in the context of clinical work. And you know that currently the recommendations of the American Heart Association state that we have to have some amount of omega-6 in the diet, and there was also an estimate of the intake. And the basis of this recommendation is randomized clinical trials for omega-6 PUFA supplementations. And there have been repeated statements by many authors of these recommendations that most of the evidence for the beneficial effects of omega-6 come from randomized clinical trial evidence. Now I want to draw your attention to this paper that was recently published in the British Journal of Nutrition that challenged the evidence for the beneficial effect of omega-6 in the diet, testing whether N-6 specific PUFA diet increase or decrease the risk of coronary heart disease events in randomized clinical trials. The method was an accurate search and scrutiny of the literature examining the relationship of omega-6 with the coronary heart disease in randomized clinical trials, really dwelling on each single trial. Here are the 9 trials that have been investigated this issue, but if you see whether the trials were really related to intake, increase the intake of omega-6 fatty acids, or were a mixed intake of N-6 and N-3, you see that only a few trials really were specific for N-6 fatty acids. And the conclusion, which is also my conclusion, the conclusion of this paper is that when you isolate the trials that substitute N-6 PUFA without simultaneously increasing N-3 PUFA, you have an evidence in randomized clinical trials for an increased risk of death that approaches statistical significance. And this is probably the reason why the advice to specifically increase N-6 PUFA based on the data of this randomized clinical trial is likely to increase cardiovascular disease and coronary heart disease risk. So the overall conclusion is that there is a convergence of epidemiological, animal and in vitro mechanistic data proving that omega-3 fatty acids are potent anti-inflammatory and anti-atherogenic nutrients. The role of omega-6 fatty acid is controversial because it depends on the comparator, whether you're talking about competition with saturated or with omega-3 polyunsaturated fatty acids. And from the practical standpoint of giving dietary advice at the population levels, to me makes no great sense to recommend the simultaneous intake of omega-3 and omega-6 fatty acids. And I leave you with these considerations. Thank you.